Hi, welcome to Leo's biology class. Do you ever wonder how cells grow? Well, cells go through a process called mitosis, where cells actually duplicate themselves to create exact copies. So let's talk about mitosis. Mitosis helps us grow taller because adding more cells can help us grow. Now, this is on the left is Victor Wembanyama, 7'5", standing next to a 6'7" Reggie Miller. So clearly, these two have undergone a lot of mitosis in their life to help them grow as tall as they are. Now, not only does mitosis help us grow taller, it helps us heal. So if you ever get a wound, mitosis can help repair the damaged skin, the damaged cells in that wound to help us heal. So cell division has two parts. In the blue, we can see interphase, which is going to be the prepping for the later stage in red of mitosis. Interphase is the vast majority of a cell's life. Mitosis can be split into four parts: prophase, metaphase, anaphase, telophase, or also known as PMAT. It's a good way to remember it. Prophase is by far the largest of the stages and is also the first stage. Because of the size of prophase, we're going to see prophase split into two general phases of prophase and prometaphase. Now, prophase and prometaphase have no real necessary boundary, so there's no saying of when prophase ends and prometaphase begins. Okay, so during prophase, we're going to see three major things happen. First of all, where the arrows point, we're going to see the chromatin condense into chromosomes to make them easier to pull and move around the cell. We're going to see the spindle fibers start to unwind, and these are going to be later used to organize the chromosomes across the cell. And then finally, we're going to see the nucleolus break down to enable all of these chromosomes to move around the cell in later phases. Now we're on the pro-metaphase, and we're going to see three things happen. We're going to see the nuclear envelope finally break down, enabling the chromosomes to be able to be moved. We're going to see the spindle fibers grow more, some of them are going to connect to the kinetochores, which are proteins at the centrosomes of the chromosomes, and that's going to enable them to be pulled to either side. And we're also going to see these the chromosomes finally condense, fully condense, after we saw them starting to be condensed in prophase. Okay, so now we're going to be entering into metaphase. In metaphase, we're going to see the chromosomes line up at the metaphase plate because the spindle fiber is going to attach to the kinetochores and push all the chromosomes to the center. This middle is going to be the metaphase plate, or the equator of the cell, and it's kind of an imaginary line where all these chromosomes meet up. And the idea is that when these sister chromatids are pulled away from each other, we're going to want to see an even number in each of the daughter cells. Now we're going to enter anaphase. Anaphase is very simple. The sister chromatids are going to split apart, and the microtubules are going to push away from each other and pull the sister chromatids to each poles of the cell, so that each daughter cell is going to have an equal number of chromosomes. And then we're going to see the cells start to slightly elongate to enable that division later on. So now we're entering telophase. Telophase is kind of trying to get the cell to go back to normal. So in telophase, the cell is nearly done dividing. What we're going to see is the mitotic spindle that's been doing all this stuff for the last couple phases is now going to disappear. The nucleolus is going to reform, and then we're going to also see the nuclei reform again, and the chromosomes are going to be decondensing from that chromatids to the chromatin, which is the stringy form. So now we're going to be entering cytokinesis, the final stage of cell division. And what we're going to see is that right in the middle of the two daughter cells, a cleavage furrow, which is right between, is going to kind of pinch in and separating them so that each daughter cell is now going to be independent of each other. So plants do this actually a little bit differently because they have a stiff cell wall. What they're going to do is build a cell plate in between the two daughter cells. So what's going to happen is the daughter cells are going to be separated but still stay attached, while the cytokinesis in animal cells is actually going to divide them, and they're going to be completely independent of each other. So interphase, which is where the cell spends the majority of its life, is also going to have the duplication of the chromatin and the duplication of all the organelles. Then we're going to do PMAT to remember everything from prophase, metaphase, anaphase, telophase. Prophase, you're going to be prepping. Metaphase, you're going to be meeting at the middle. Anaphase, you're going to be apart. You're pulling the chromatids apart. And telophase, you're finally going to be separating. So you can remember that as PMAT. So remember at the beginning of the video, I explained that interphase is the majority of a cell's life? Well, understanding interphase is critical to understand how mitosis can happen. Well, interphase has three parts: G1, S, and G2. In G1, we're going to see the cellular contents, excluding the chromosomes, are duplicated. This is talking about your membrane-bound organelles, which are going to be duplicated so that when the cell splits, each daughter cell is going to have an equal number. Then the chromosomes are going to be duplicated so that when you are splitting the daughter cells, each daughter cell is going to have an equal amount of DNA. Then finally in G2, we're going to see the cell double checking the duplicated chromosome for any errors and making the last minute repairs. This is critical because if you start to duplicate a cell that has misaligned or mutated DNA, and that DNA ends up coding for something that prevents the cell from receiving any kind of signals on how to stop dividing, that can lead to cancerous cells because those cells are going to constantly be growing without being able to be told to stop. And if those cells constantly are growing, that forms tumors, and that's what causes cancer. Okay, so there's also going to be checkpoints during interphase and mitosis. So starting with G1, the G1 checkpoint, which is where if you go through the G1 checkpoint, your cell is almost certainly going to divide. This checkpoint is checking for cell size. Is this cell big enough? It's going to check if there's enough growth factors, if there's enough nutrients, and if there's any DNA damage. And now, if these fail the checkpoint, two things will probably happen. One is either going to go to apoptosis, which is controlled cell death, so the cell is going to be kind of killed, so it can't reproduce. And then or it could enter the G0 phase, which is kind of the resting phase, and it's where cells just like they do their functions, but they won't divide. Now, some cells will go into that phase and they'll be fixed and they'll come out of it, but the majority of cells that go into G0 will not come out and not divide again. Then there's the G2 phase. The G2 phase is very simple. Checks if the DNA was replicated properly during the S phase, and if so, it enters mitosis. Now, the final checkpoint is going to be the spindle checkpoint during mitosis, and it pretty much checks if the chromosomes were aligned properly at the metaphase plate so that when they are separated into each daughter cell, that each daughter cell will eventually have the same amount of chromosomes, because if they weren't aligned properly, then one daughter cell will get too many chromosomes and one daughter cell will get too few. So those are the checkpoints. So that's all I have for today. So have a great day, Built-in High Wildcats. And that was MITOSIS Explained with Leo Waldbaum. Thank you for watching.